This method using the 22C3 antibody concentrate to determine PD-L1 expression in both tumor tissue and cytology specimen will expand the ability of laboratories to assess selection of patients with non-small cell lung cancer for immunotherapy in a reliable and reproducible manner. The main advantage of this technique is that it's highly concordant with the gold standard assay and will support reliable, high-quality PD-L1 testing across regions worldwide.
In general, pathologists new to this method will struggle because of the granular staining occasionally observed. Moreover, the staining of the human cells is somewhat more intense than that observed with the gold standard. Therefore, training of pathologists is necessary to obtain correct interpretation of PD-L1 staining with the 22C3 LDT.
Demonstrating the procedure will be Mrs. Marame Hamila, a technician from my lab.
To begin, fix the tumor tissue in 10% neutral buffered formalin in a cassette and embed the cassette in paraffin as outlined in the text protocol. Embed the infiltrated tissue inside a mold that is filled with molten paraffin. Let the tissue stay in the mold until the paraffin has solidified. After this, use a microtome to section the paraffin-embedded tissue at a thickness of 3 micrometers. Transfer the paraffin ribbon to a positively charged glass microscope slide. Then, dry the slide for 1 hour at 37 degrees Celsius. First, collect the bronchial washings in a preservative.